really excited to be offering and hosting the Wolbachia Project through Penn State. It is an outreach endeavor that kind of started from our research, where we look at the association between Wolbachia and its arthropod host. About 20 years ago, a group of scientists and outreach professionals and educators got together and wanted to bring this experience to high school students, not only as a way to teach high school students about molecular biology and biotechnology, but also to help the scientific community. So over the past 2 years at Penn State, we have worked with over 200 teachers. That equates to over 10,000 students that they represent. And I would say about half of those are high school, half are undergraduate. At the undergraduate level, we work with a lot of community colleges and minority serving institutions because we really want to get this opportunity out to students that wouldn't otherwise have the chance to do molecular biology. We work with teachers in 45 states across the U.S., and we have partners in about 20 countries. Wolbachia is an obligate intracellular endosymbiont. Basically, that means it is a tiny little bacterium that lives inside the cells of its arthropod host. Arthropods being insects, spiders, mites, crustaceans. So within the cells of these arthropods are these tiny little bacteria. So we figured, what if we recruit an army of students to help us kind of look at the arthropod biodiversity and determine whether or not they had the Wolbachia endosymbiont. A student would first look into their community and think about what types of arthropods they want to explore. They will look at the biodiversity of the arthropods in their community. They will go out and collect arthropods. They typically end up being common insects. They bring them back to the lab. They extract the DNA. They then amplify the DNA using PCR, or polymerase chain reaction. And then they load those PCR products onto a gel for gel electrophoresis, and that will allow them to observe the DNA and identify whether or not their arthropod has Wolbachia. Then a lot of students stop there, but we encourage them to go on to bioinformatics, where they can send us the DNA for sequencing, and then they can learn a few common bioinformatics techniques to identify their arthropod and also identify their Wolbachia sequence. The project was actually started to enable molecular biology in the classroom in a time when this equipment was not available. We're seeing more and more teachers have this equipment available, but we, through support from the One Health Microbiome Center at Penn State, are able to provide free reagents that are specific to the project. So teachers will reach out to us. They can get free PCR primers, free PCR controls, free arthropod DNA as a positive control. And then we also have an equipment loaner program that was provided by our partners such as MiniOne. And we also have other partnerships through QIAGEN, where they will actually provide free DNA extraction kits. So by forming these industry partnerships, teachers can then get most of the supplies for free.